Alrighty, good morning everyone and welcome to Symbio's technical webinar. I'm Christian Just, I'm the National Sales Manager at Symbio, and today we'll be hearing from a couple of our technical experts on the topic of Shigatoxin lab testing and results. We've got a fairly good turnout to today's webinar, so we've got supermarkets, food manufacturers, government bodies, universities and other laboratories. So really happy with the diverse range of stakeholders and really interested in the questions that we might get from different angles at the end of the presentation. So we've got the Q&A bar just above in the Teams meeting. So if you just want to click on that and enter your questions, one of our technical team will be able to answer at the end of the webinar. Alrighty, so just before we start off on the technical talk, there's a few attendees that may not be familiar with Symbio. So I just thought we'd give a brief introduction into who Symbio is, just so you can get an understanding of the context of who we are in this testing. Alrighty, so Symbio, a bit of background. So Symbio is a national laboratory. We've got laboratories operations in all the major mainland cities across Australia. We offer an extensive range of micro and chemistry and residue testing, and we are involved in the fields of food, agriculture, environment, and pharmaceutical testing. With Symbio, we also have an extensive range of accreditations. All of our laboratories across Australia are ISO 17025 accredited. We're also a government-approved lab for importing and exporting. We hold a government contract for the NRS, so that's testing a lot of the commodities for pesticide testing. We're a quarantine-approved premise, TGA and ABVMA licensed, amongst many others. So all that accreditation just goes to demonstrate the robustness of our quality management system and making sure that we issue correct results, so you've got confidence in the results that we issue. So the difference we try to provide our customers with the value is we're the largest independently owned Australia with labs across Australia. So with all the different labs, we've got contingency. If there's any breakdowns, we've got backups. All of our laboratories are equipped with the latest technology, with instruments and also operated by expert staff. We also offer IT solutions that we can customize for our customers. We offer very competitive and transparent pricing, and we're dedicated to R&D to ensure that we're always coming up with the most up-to-date methods to make sure that we minimize turnaround time. Turnaround time's our main focus, to get the lowest turnaround time possible to make sure that you're getting results in a timely manner. Okay, and so we just thought we'd do a bit of a virtual lab tour, just so people can sort of see our operations. Just a couple of minute video, so I'll just get going. One sec, I'll just turn that volume down. Yep. Okay, so this is our laboratory at 8 Mile Plains in Brisbane. All our laboratories have the similar setup, with chemistry on the top floor and microbiology on the lower floor. So at the moment, the drone's flying over our wet chemistry area upstairs. So this is where we test things like food, agriculture, environmental samples, and all of our pharmaceutical testing is done in a building next door. So it's separate to our food, agri and enviro testing. We're about to fly into our instrument room. So this is our GC and LC room, so liquid and gas chromatography mass spectrometry. This is where we test things like pesticides, mycotoxins, PFAS, vitamins, hormones, many, many others. And you can see we've got multiple instruments. So if there's any breakdowns, we've got that contingency and we can run on another instrument. Back on the wet chemistry floor, we're about to fly into another instrument room. So in this first bay, this is for protein testing, so combustion. We've got sugar profile testing, fatty acid testing, profile testing, amongst many, many others. All those instruments down the back there are environmental GCs and LCs. Now we're in our allergen room. So this is where we test multiple different allergens, I think up to about 20 different allergens. And there are ICPs there that test all the metals, both to high levels and low levels for your things like cadmium, mercury, and lead. So that's pretty much upstairs of the chemistry floor. We're about to go downstairs into our micro lab. And here you can see, so very, very open space. So all these benches, we do all of our plating. We've got stomachers. We do microscope work. All on the right-hand side there was all our incubators, multiple different temperatures for all the growth. And this is our login area. So each bench has its own set of commodities. So food, environmental samples, water samples are all separated. There's our fridge for storage and retain samples. And now we're flying over to the PCR and multi-top rooms. You don't really get a good look at it, but this is where PCR happens. We've also got our robot there that does a lot of the streaking and plating. And then that was our microbiology lab. So just to finish off, we'll just quickly fly past the rest of the building. So here we have our customer service team. So our customers may have talked to some of the team members here. And then just into our training room. So Symbio takes training very, very seriously. For all of our accreditations and making sure lab staff are trained in all the new methods, this is where some of the training takes place. So again, this was our Brisbane lab. It's very, very similar to our Melbourne lab. However, Melbourne is, I'll just finish this, sorry, and show. So Melbourne is our largest laboratory. It's based out at Ravenhall. So everyone, please, if you're ever in the area or want to do a lab tour, you're more than welcome to get in touch with us and we can arrange that. So look, that was a bit of a brief outline of Symbio, but without further ado, I'll get on to the technical slides, which I'm sure everyone's here waiting for. So I'd like to introduce Dr. Loveleen Kaur, who is our technical manager in Micro.
All right, well, thank you, Kristen, and good morning, everyone. As Kristen said, my name is Loveleen. I'm the technical manager in microbiology at Symbio Laboratories. I've been with Symbio from past 4.5 years now. I've been working in commercial testing industry sector in Australia and overseas from past 16, 17 years now. And I'm also an approved food analyst and a technical NADA assessor. So today, considering we have got a very wider audience from different sectors within the industry, we will try not to use very technical terms. However, we want to touch base on everything starting from when we first introduction to E. coli and STEC, and then when we receive your samples in our laboratory, what happens with it? Because considering with the past few weeks, we had a lot of questions from clients and from other sectors asking to help in understanding the test results, the test report, the methods, etc. So that was the main idea of this forum, that we will take you through when the samples arrive to us, what do we do, how do the reports look like. So we'll be covering all different aspects today. At some point, I will also have our operation director, Kashif Ahmed, joining us as well. So he will have some inputs or insights into the into the testing that we perform. If you have any questions, like Kristen said, please keep it towards end of the thing. We are always there to assist you. We can always take all the questions at the end, and you can reach out to us when required to. So next slide, please, Kristen. So just to start off with a very, very basic introduction of E. coli, majority of you may be already familiar, but some of our guests today may just need to go take a step back and first try to understand what exactly E. coli is. So if we look at the presence of E. coli, when we are talking about a hygiene indicator, so E. coli is one of those organisms. So when we are doing, could be our premises cleanup or for our food safety, or even for your staff hygiene, you're looking at E. coli as one of the routine indicator. But because it's a very diverse group, it's a big group of E. coli. There are so many different serotypes, different serogroups present within E. coli, and it's a very, very widely distributed organism, and it's present everywhere. You can take some water sample of a creek, and although in the drinking water you don't want to see E. coli, but there are a lot of environmental E. coli that you will find there as well. You look at a soil sample, you will get it from there. But the interesting thing is they live with us in our intestine, in a gut as well. In normal human beings, they are present, and in some slaughter animals and others, the pathogenic ones are present as well. So most of these strains, when they are living in our gut, they are perfectly harmless. They cause no harm to us. In fact, they are more of a friendly bacteria They help in production of vitamin K, which you need for your blood clotting. They also take up oxygen, so that means they are not letting the bacteria which need to use oxygen to grow. They also help in digestion at some point. And if you just look at the human gut flora, which is full, the lining is full of all different microbes. E. coli itself is about 100 million cells present in there. That means, like, 0.2% of your total gut flora is actually E. coli. So it's not that bad as it gets portrayed at different times. Next, please. Now, when we are looking at E. coli, just like us human beings, we, in a society, you will find good, bad, all kind of people. So this is how E. coli is. So there's a good E. coli and there is some unfortunately bad E. coli serogroups as well. And how do we differentiate? Like, what makes them good, what makes them bad? And I see it again, just like, us, it's the genetic makeup. Some of the good genes, they don't let them do anything wrong, and some of the bad E. coli strains will have some genes in their DNA which will make them the bad ones. Now, what does the bad genes really do? So if we look into a genetic system, when we have these proteins, or we get infected by diseases, it all means that our DNA is encoding certain genes. Those genes are then triggering the production of something which is harmful for us. In this way, the pathogenic E. coli, they have got some genes. With now these genes trigger the production of toxins. And when these toxins are produced, they make these strains harmful. Now, what happens with the toxins? So toxins is more like a protein, which is a bad protein. It's like chemicals. They get injected into, when these are produced, they go into our gut. When they go into the gut, they then get attached to the gut, or they cause breakdown of the lining of the gut, and this is where the normal functions of your body get affected. And when this gets affected, this is where the illness comes in. Now again, although you at some point we may have some bad E. coli in us, but it all depends if it end up in the wrong spot. And also on the immune system. So some of us may have very strong immune system, and we may be carriers of the bad E. coli, but we will never have this illness because our immune system is too strong to fight this bad E. coli. So even the presence of the bad E. coli doesn't really mean that you will fall ill, unless you are from that category, which is could be immunocompromised patient, with the age, or children. This is where that your body will then decide that even if you have bad E. coli, is it going to cause any illness? And one of the most important pathogenic strains, which is identified, is called STEC, that is the Shiga toxin producing E. coli. That means this particular group of E. coli produces toxins, which are the Shiga toxins. We will discuss more in detail what Shiga toxins are. And this is the kind of group that we'll be discussing today, called STEC. Next, please. Now, when we are looking at toxigenic E. coli, or the STEC group, as I mentioned before, when you have these E. coli and enter into your gut, into your intestine, what do? They will obviously find somewhere to sit down or make a home, and they will harbor to your gut wall. Now, once they attach themselves to the gut wall, obviously they come into action. Now they are very comfortable there. They will do what they are meant to do. This is when they will break down your gut wall. They will make holes in that, and this is where you will start feeling ill. The illness could be as simple as you may have diarrhea for 1 or 2 days, or could be even worse when they have produced Shiga toxins, and now the toxins have entered your bloodstream. So once the toxins enter bloodstream, this is where the things get really, really serious. Now the toxins can reach to any part of the body. It could be to your kidney, and this is where it will cause more harm than normal diarrhea. And this is where things start getting more complicated. Now, if you have sugar toxic, if you have ingested food, or somehow the toxic E. coli got into your body, you're not going to see the effect immediately. It's not that you had your food and you will start feeling sick after half an hour, 1 hour. It may take 3 to 4 days for these symptoms to appear. So this E. coli will first find the right place in your body. It will produce the toxins, and it may take even longer than 4 days. So this is not an immediate effect. So the diagram in this slide, the figure that you see, is giving us an idea that where does the STEC sits when we are talking about E. coli or the wider group of coliforms in here. So when we talk about coliforms, it's a very big group, and within those total coliforms is, again, as I said before, they're used as hygiene indicators in food industry, in environmental industry, in water sector as well. So when we are looking at total coliform, which is a very big group, within that this called fecal coliform, which where origin is from feces. And then within fecal coliform, you have got a smaller group called E. coli. And within that E. coli, you have got this Shigatoxigenic E. coli, a little subgroup of that. So when you're looking at the whole umbrella, this is where STEC sits in the bigger total coliform group. Next, please. Now, talking about Shigatoxigenic E. coli in food. So where does this where does this toxins or the STEC E. coli comes into the picture? Now, if we go back a few years or few decades, the initial outbreak that was first reported was from an undercooked meat. So it was either an improperly handled food, meat, or undercooked meat where, as I said, that E. coli, you will find in abattoirs, in slaughterhouses, that it's actually present in the gut there for animals. So you will find it there. Now, after that initial outbreak, which was just related to meat samples only, after that we have seen that recent outbreaks are are associated with very wider group, and those wider groups now include raw milk, dairy products. So when we talk about raw milk and dairy product, if we look in Europe, especially in France and other countries where we are using unpasteurized or raw milk to make cheese, you will find this outbreak is pretty common in those countries. In Australia, we are not allowed to make cheese for raw milk, so we're pretty safe in here. However, you will find that dairy product where unpasteurized milk is used, because E. coli, STEC E. coli would not survive at higher temperatures. So once your process has gone through pasteurization, you have killed E. coli already, or you have minimized the risk of spreading it. Then various cooked meat, when although we talk about cooked meat, but it's more like undercooked meat. And then this could be anything from your burgers, from something coming from the abattoirs, or from the supermarket, or even at home, if it's not cooked properly. We've seen a lot of fruits, could be apples, melons. Now, E. coli will not get into the fruit. It's actually on the covering of the fruit. So it's just outside of a melon or an apple. And we will discuss more about transmission in next slide. So this is somehow this E. coli would have traveled from the farm onto the surface of the fruit, and then obviously traveled to the end user, where they would have consumed the food contaminated with that. Vegetables, same thing. It could be your spinach, your lettuce, or your salad, something that we don't cook, or we don't cook at a higher temperature, and would have come in contact with contaminated water, would carry this E. coli with it, because most of these vegetables, when you consume, you would have kept in the refrigerator, so they would not have gone through the heating process. So the E. coli is still happy there, waiting for the right host to multiply and cause the infection. Nuts and sprouted seeds were also part of the recent outbreak. Again, not heated. That's why they become part of that. So all the common denominator here is that anything that you're consuming raw, hasn't gone through the heating or the pasteurization process, is actually at a risk of carrying E. coli if at any time point of its life cycle it may get in contact with this E. coli. Next, please. Now, the current topic, STEC in leafy greens. So in leafy greens, it's very, very common to find STEC. And then when scientists were looking into it, why does it make it so attractive for E. coli to attack these leafy greens or salads? And then various researchers have come up with this hypothesis that it's the surface roughness of these kind of greens which let STEC sit on it, and also the wax coating that you apply on these, or naturally either you apply or is naturally present on these greens, will impact the risk of contamination. And when we look into lettuce, we'll see that E. coli will sit because its surface is so smooth. It gives us a better surface area. It gives us a better room to sit on it and, of course, survive for longer period. So this is why lettuce in particular have got wider leaves, smoother surface. E. coli would just sit on there and it kind of like hidden there until it find the right host for it. It doesn't have the wax coating on it, so the when it sits there, there's nothing for it to prevent or it won't fall off even when you're washing it. So this is why lettuce is one of the most preferred leafy green where STEC will grow. If you look in lettuce as compared to other leafy greens, for example, kale, which have got more rigid, waxier structure texture to it, you will find that E. coli would fall off while you're carrying it or you're washing it. It will fall off easily. So that's why you will find less cases of kale as compared to lettuce. Next, please. Now, we want to see if it comes on leafy greens or on fruits and vegetables, how does it actually, transmit from the farm to the fruit or the salad itself. So there are many ways of it. Of course, the first route of transmission is from human faces to oral transmission. This is how it travels in human beings, right? So there has to be certain hosts in between, certain vectors in between, how it travels from faces up to the end user again, or to the leafy greens or to the salads here. So if you would have used the water which is contaminated to irrigate the fields, of course this water would have E. coli in it. You bring this fruit cultivated or the vegetables cultivated using this contaminated water, you are actually adding E. coli onto it, and then when it comes to your warehouse, to your packaging facility, you're actually carrying it along with your fruit there. Anytime if you look into the soil, that you will see many farmers, they would like to test the soil, which you have seen recently. There was a lot of clients asking for testing of the soil, which is good in a way. They want to test the soil before they use it for sowing, so that they are sure that this soil is not contaminated with E. coli or human feces. So there's no risk of contamination or transmitting the E. coli or other microbes, or could be other pathogens as well. Right now, there's a trend of using a lot of organic fertilizers, which could be manure. So, of course, they haven't gone through any processing with any chemicals or anything else. So, if you had these manures came in contact with human feces, then they are carrying that. The E. coli is just sitting in a certain inactive state. As soon as you put your product in there, it will get in contact with it and it will get to your store, to your supply house, to your warehouse. And then not just there, of if you have your the farm that you're using, it's using equipment which is already contaminated, your tools in your warehouse where you're packaging, or in the surfaces. And same goes for the abattoirs as well. If you're mixing, two products, if you're grinding your meat and your tools are not correct, your mixers are not cleaned properly, you will then end up transmitting E. coli from one sample to another. Your processing facilities. Of course, as I said, E. coli is known as a hygiene indicator. So if your processing facilities are not maintaining adequate sanitation practices, you will be transmitting E. coli from your, you may be adding on to, you may get your fruit or vegetable or your meat, which was previously not contaminated, but your processing facility could be your tools or could be your staff adding on to the samples here. Then when once you have packed, once you have sent your product to the retailers or to the wholesalers, the mishandling can occur during transportation. So if you have your transport vehicle carrying your raw meat next to your fruit veg and they are not packed properly, there's no right segregation, this is called mishandling, and you will be then cross-contaminating the samples. And one thing which you may not have much control on is your end user, your consumer. So when they buy things off the market, they bring it in the fridge, they may unpack your raw meat, put the salad next to it, use the same chopping board, don't wash their hands in between. So this is where they will get cross contamination in the kitchen as well. Now, we mentioned in the last webinar as well, DPI New South Wales have issued a document which is very, very informative, very easy to understand, which will also help you to pass on to your consumers to educate them how to control STEC cross-contamination there. Next, please. Now, STEC serogroup. So again, remember we mentioned that E. coli has got many different serogroups in there. Now within STEC as well, so STEC is one of the group within E. coli. Within STEC, we have got multiple serogroups in there. So about 470 or even more serogroups are very well known and studied. But when we are talking about human illness, something that's been identified for public health awareness or something that causes risk for public health, there are big 7 or top 7 STEC serogroups that are taken into consideration. So among the wider group of serogroups, only 7 groups are the one which are directly linked with human illness globally and in Australia as well. Now, when this is a very common terminology, so when you're requesting a test with SymBio or any other laboratory or even your regulatory bodies, they will be saying, oh, we need to test for STEC 7 or top 7 or big 7. So by that, what they mean is that you need to test for E. coli O157, which is one of the groups of the top or big 7, and then the rest of the 6 are called non-E. coli O157. Now, non-E. coli O157 covers rest of the 6 serogroups, and they all O157 and non-O157 makes the big 7. Now, for the abattoirs, especially if you're sending your product for under meat export program, depending on which country you are sending it to, some countries will just need E. coli O157. Some countries will need all the big 7. So depending on your regulatory requirement, you will test for that. For leafy green vegetables, again, because of the current risk that we were trying to mitigate within Australia, it's important that we're looking for all the big 7 so that you are covering the risk of any Shigatoxigenic, which is currently classified as a part of public health. Now, Meat Livestock Australia has got abbreviations very well explained. So the abbreviations are just taken directly from there, and you can always refer to their document for more details. Next, please. Now, when we're looking at Shigatoxigenic E. coli in food, as I mentioned before, that O157 is one of the group which is most widely present. So this bar graph is from one of the global publication, and this just to give us an idea that whenever there's an outbreak, one of the most widely causing organism is actually E. coli O157, and it actually comes from the raw crops as well. So others like beef, dairy, fruits will have it as have the outbreaks as well. But O157 is the major dormant serogroup which is related to most of the food recalls or food STEC outbreaks that we usually get noticed of. Next, please. All right, so this one is a study directly performed in Australia from a New South Wales group from the universities plus some other researchers in the Department of New South Wales as well. So in Australia as well, just as in globally, O157 is the main STEC serogroup that has been identified and linked to various food recalls. Now, when this group tried to sequence the E. coli STEC that has been identified in Australia so far, when they sequence that, O157 formed 35% of the isolate that's been isolated or that's been serotyped so far. So when we look at the other non-O157 group, there are 6 serogroups in there, but out of the 6 serogroups, only 2 of them, O111 and O26, are the ones which are found in Australia. Other 4 serogroups from non-O157 group, they are rarely reported in Australia. So if your lab have issued your report with these 6 serogroups which are not currently found in Australia or are very rarely reported in Australia, it is important that we go back and check if everything is okay with it, because this will definitely need further consideration. That is it, like you have got a right to question how it's formed. Maybe we need to involve more research groups like CSIRO to help us understand where it might have come. The problem could be much bigger than you would have anticipated by just finding those serogroups. So if you get a test report with O157, O111, and O26, they are the common types in Australia. But if you get a test report which has got other 4 serogroups, then we need to find out that they are not commonly found in Australia. Where are they coming from? Next, please. Now, this was just to give us an introduction before we head off to laboratory testing of Shigatoxigenic E. coli. So from this part, I will be handing over to my colleague, Sarah. Sarah is a lab manager in SymBio, and she will take us through the life cycle of the test sample once it arrives in the lab. Over to you, Sarah.
Thanks, Loveleen. My name is Sarah. I'm the laboratory manager of the microbiology laboratory here at SymBio Laboratories. I have been working in the food and environmental testing industry for the last 20 years, working across a number of different NADA accredited laboratories. I have been here in this role as laboratory manager for 8 years for SymBio, and I'm responsible for the day-to-day operations as well as the STEC testing, so the preliminary screening followed by the confirmation. Thank you. Next slide. So these are the 2 methods that that is available for screening for top 7 E. coli 157. So the method, the USDA MLG5C method and the Assurance GDS methods. Now, these are both PCR-based methods. They both have accreditation from different industries. The MLG5C is accredited by the USDA. It's also NATA accredited and DAF approved. And the Assurance GDS method has AOAC approval, also NATA accredited and DAF approved. They both test for the big 7, so E. coli 157 and top 6, and they can also test for E. coli 157 on their own. And these 2 methods are available at all of our SymBio sites with a 24-hour turnaround time for the screening test. So what is a screening test? A screening test is where we first do the preliminary testing and we end up with a result of either not detected or potential positive. And I'll go on in my next slides and explain that process. Thank you. Next slide. So the initial screening test that is performed is a PCR test. So this is where we use DNA and we amplify that DNA to a detectable level. What we're actually looking for in the DNA is the virulence genes, the STX and the EAE gene. These are specific to the STEC E. coli group, and that is what the PCR is amplifying. This is commonly known as a screening test because we are not giving a confirmed positive result at the end. It will be confirmed negative or not detected or a potential positive. So the basic overview of the process would be that we would weigh up the sample. So we would take a portion of the sample and prepare it. This might be 25 grams or more. We add what's known as a selective enrichment, and we incubate that overnight to grow the bacteria. And then we perform, after the bacteria has grown, we perform a PCR step, which is the initial screening. And from that PCR, we have a negative result or a positive. So the negative report would be reported as not detected and no further action would be required. But a positive PCR result would be reported as potential positive, and further cultural, biochemical and genetic confirmation would be performed. I'll explain this in detail in my next slide. So as we move through, thank you. So the sample preparation step is a very important step. This is where the sample would be submitted to the laboratory. Usually it is submitted in the retail packaging, but this is not always the case. Sometimes sample sizes are large and they cannot be submitted in their original packaging, or subsamples are taken from various lots throughout the production and sent to a laboratory as a composite. So we can see in this example here, we've got a crispy leaf salad, and it comprises of different various types of lettuce. So when the sample is taken, it is important to take what's known as a representative portion. So different pieces of that lettuce would need to be taken in order to make sure that we are capturing or potentially getting an opportunity to see a representative sample and actually recover the bacteria if it's there. We might also receive raw meats as well, and these raw meats may come as a drilled meat, so they won't be in that representative retail packaging. We may also get a juice. So in a juice sample, we would need to make sure that it is mixed thoroughly before we take that, because the pulp will concentrate the bacteria down the bottom of the container, and we need to make sure that we mix that thoroughly before taking the subsample. When a subsample is taken for testing, it's usually taken as a 25-gram sample. This is the Australian standard for most pathogen testing, not only E. coli, but also things like Listeria and Salmonella. And then that 20, but in some industries, it's multiples of 25. So there might be a 5 by 25, so a total of 125-gram sample, or even more than that, 10 times 25 grams as well. When we take that representative portion, it's placed into a sterile bag. This sterile bag is free from bacteria to ensure that there's no risk of cross-contamination during that sampling process. Next slide, please. So this is, so once we've got the sample in the sterile bag, we need to grow the bacteria. Now, to grow the bacteria, we use something known as enrichment broth. This is basically a liquid form of food for the bacteria. It contains different things like proteins, carbohydrates, sugars, minerals, nutrients, everything that the bacteria needs to grow. It is important that that media or the enrichment media that we're adding is sterile. So prior to us adding it to the bag, it actually is sterilized at 121 degrees for 15 minutes before it is added into the sample to ensure, again, that there is no risk of any additional bacteria being cross-contaminated into that sample. So the result at the end of the analysis is true for the sample that we've received. Once the broth is added to the bag, we use a homogenizer to mix the sample and crush it up. The homogenizer works almost like a stomach, where we have two paddles and it's pushing the sample around, breaking it up, making sure that we are extracting all the bacteria, whether it's on the surface or within the food, so that it actually has the potential to grow overnight in incubation conditions. Next slide, please. So once we've prepared the sample and the broth has been added to grow the bacteria to really, really high levels so it's detectable, that sample in the bag is then placed into an incubator. So this incubator is a temperature controlled room or desktop sort of version. And in that, it has that time overnight to actually grow. So all the nutrients from that broth that we've added will help that target bacteria to grow, but also the temperature that we would incubate it at, which is 42 degrees, is a temperature that E. coli loves, but other bacteria does not like to grow at. So by putting it at 42 degrees, we're giving the best possible chance for E. coli to grow, but also reducing the chances of other non-target bacteria to grow during that period as well. So once we've received your sample, the incubation takes around about 18 to 24 hours. Once that incubation period has completed, we start the PCR process. So that enrichment bag with the sample and the broth and the bacteria, which has grown up to millions of a level, that's taken out, and an aliquot is taken from the bag after mixing and put into a tube, and we start what's known as the DNA extraction process. This is where we concentrate the sample by centrifuging, and then we start to extract the DNA if E. coli is there. And then we go through the process of PCR. So the prepared sample is put through PCR, And if there are the target genes, we will see what's known as amplification. If we can go to the next slide, please. Thanks. So once it's gone through the PCR and there are multiple stages of amplification, if the genes are present, we will see what's known as a curve. So we would be expecting a curve for the gene STX as well as the EAE gene. These curves need to amplify above what's known as the threshold line. So there's a grey line there that says threshold, which is dotted, and we can see that we have a curve for the STX1 and STX2, which is coming up over that line. That will produce what's known as a positive result. If there is no amplification because the genes are not present, we'll get what's known as a negative result. So we can see down there the green line where there is a sort of a little bit of a rise, but nowhere near the threshold. This is what would be deemed as a negative result for PCR. So when the laboratory has a positive result, we're expecting the curves. We also run controls. So we run DNA as well as live bacteria through this process, and we can see that those have worked and the curves are typical for those. So we know that the PCR reaction has worked. So a negative is a true negative. But if we have the actual curves on the sample because the STX and EAG are present, we would call this a positive PCR. This is positive on the PCR test, but it doesn't mean that the sample is actually positive for the STEC E. coli. It does require further cultural, biochemical, and genetic confirmation. Next slide, please. So when we're interpreting our screening results from the PCR, if we would consider a negative result if the XTX and or the EAE gene are absent. So we're not seeing any amplification for those genes. What we would consider a positive result for a PCR screening is both the STX and EAE gene are present, meaning that we will see the curve. We must make a point that this is not the final result. This is what we call a potential positive result. The way the PCR works is that it does amplify genetic material, but if that genetic material isn't from live bacteria, we will still see a curve. So we must realise if this is only a potential positive or presumptive positive, meaning it may be positive or it may be negative. It allows, when we release this result to clients, it does give the clients some option at that stage to either put their product on hold, downgrade their product, or take any action that they feel would be necessary at this point based on the presumptive positive result. It is important that full action is not taken until the confirmed result because there are possibilities that a presumptive positive result will end up as a not detected also. Once we've notified a client of the potential positive result, the usual turnaround time from this point, the screening test, to the final confirmation stage and the final report of not detected or detected usually takes around 2 to 3 days. Slide, please. So this slide here is showing how those screening results would be reported. And this is what most of the food clients would be seeing when they get it. They would see a not detected or ND. So this is where we have a negative result on the PCR, but the way that that negative result is reported, it's not reported as negative, it's reported as not detected. It will also show on the report details of the sample, the methodology used, and also the unit. So in this example here, there was 375 grams tested. If there was only 25 grams tested, that would be the unit that would be listed on here as well. Once a manufacturer receives this report with not detected, no further action is required. And the usual turnaround time for a screening report of not detected would be 1 day. Next slide, please. This is an example of a report where we had on the screening test a positive PCR result. Even though it's considered positive on the PCR, this result is reported as potential positive. That's because we need to do further confirmation needs to be done to determine if the bacteria is live and culturable, or if it is a false positive result and there is something else in the sample that may be producing that curve and producing a false positive result. This again will show the methodology used. So in this case, it was the MLG5C method. It will show the units that was tested, 375 grams, and the word potential positive. This report would be issued as a preliminary report, but not as a final report. And when the final results come through after the confirmation, 2 to 3 days later, that is when the final report will be issued with the either not detected or detected result for the different STEC groups. Thank you, Loveleen. I'll hand back over to Loveleen now. She'll go through the steps of the confirmation. So after that screening test has gone through and we have the potential positive, what happens to the sample from there? What are the steps that are taken and how do we come to that final result?
All right, well, thanks, Sarah. So Sarah has covered the primary part, what is called screening of Shigatoxigenic gene now. So this kind of testing is very common in industry, and many manufacturers, abattoirs would have their own labs also performing this testing. So because this is all that you need to do here is get your sample, enrich it overnight, screen for the Shigatoxigenic genes, and then see if the result is negative. You just, no action required. And if the result is positive, this is where it requires confirmation. Now, when we talk about confirmation, so what we're trying to say is that to confirm that the results produced during screening are actually confirmed from coming from a viable E. coli. Next slide, please, Kristen. Now, this confirmation testing, why I mentioned that screening testing is performed by majority of the laboratories. Many labs are accredited by in-house laboratories, are accredited by NATA to be used as in-house laboratories in abattoirs, in manufacturer site as well. But when it comes to confirmation test, it's a very complex multi-step testing. It involved many reagents. It involved different techniques in there. So the laboratories need to maintain that competency, that reagents, and then also be able to produce this testing, which will take 2 to 3 days or even more if we have to keep going back. Now, I'm going to take you to the different steps of confirmation. This may sound very technical at times, but we can, as I said, we can always keep the questions till end. I'll try to simplify the language that we are using in here so that everyone is on board and can get it what we are trying to say. So in confirmation, it involves multiple steps. The first step is before we can start anything, once we have a PCR positive, we go back to the sample bag which Sarah has shown before, where your sample was enriched with the liquid food for letting the bacteria to grow. So that bag is now called a bag with enrichment broth, and it's called now positive enrichment broth because the PCR result for screening has come back positive. So the first step for confirmation is now from that positive presumptive enrichment broth. We will take that broth, and that broth has got many bacteria which have given you a positive PCR result. It is assumed that now those bacteria were given the right condition to grow in there, and there will be many of those cells sitting in there which we can now take. And now before you can do confirmation, you need to see this bacteria on a solid agar or media before you can do any other testing onto it. So you need to then streak that bacteria from that positive broth onto a solid media agar. Now, solid media agar is a selective food for E. coli. So here we use specific media which allows only E. coli to grow on it. So if you assume if the sample is coming from, say, for example, abattoir, it will have lot of other background flora. By background flora, we mean many other microbes apart from STEC or generic E. coli growing in there. But you are only interested in this testing for STEC strains to be isolated. So you will use a specific media which will only allow STEC strains to grow on that. That's why those media plates are used. We streak this broth onto it, and then it gets incubated for a certain period. After incubation, we look onto the plates, and see if this we are able to get the isolate. So this is the first step of confirmation. Now, mind you, in this whole process, there are multiple steps which can help us take certain decisions, because if your first step doesn't give you any isolate, you cannot continue to the next step. That means the bacteria which produce the genes which you identified during PCR are not coming from the viable organism. It could be when you did your washing step, your washing during the packaging step, the bacteria may have died. So the only genes that got detected are from the dead bacteria. So when you try to isolate that, and if the sample, if your bacteria is not present, you have to stop the test there and release the report as not detected. So this is our first step, and it takes 24 hours for us to realize if the bacteria is present or not after incubation. So the testing may just stop here, and we can just report as not detected if there is no viable bacteria growing onto the plate. Now, let's assume that the bacteria was able to be isolated. It did grow onto the plate. What next? So then we try to see, okay, now this isolate that we have, we need to identify it using different biochemical steps onto it. The first one is called antigen screen. Now, O antigen screen means it's like antigen antibody reaction. So when we, COVID is a good example here. When we used to have those lateral strips, we will put a sample, nasal swab sample or something, and then you will see that line coming up in there. So this is the same antigen antibody reaction, that you had antigen from your sample and antibodies in that lateral strip. You let it react together to give you a visual reaction like a line appearing on the strips. Same way, in this antigen screen, we add antibodies, and then antigen is from your sample, and we try to see if they agglutinate. Agglutinate means they form certain bonding, they form certain clumping together. So that's very visual. We can look into it, and if it doesn't, that means it's not the E. coli that we, a serogroup that we are looking for. That's the first step. Then there's a second step called immunomagnetic separation. This is a kind of purification along the same lines. We use a different, it has got specific reagents and kits that you have to use for this process. And then you make it as a part of, again, screening process for the right O serogroup. If your O antigen screen or your IMS does not give you the right positive reaction or a typical positive reaction that you're looking for, the result can be concluded as negative at this stage and reported as not detected. So coming back from the isolates where you were looking at typical phenotype, by phenotype you mean how the isolates, what the isolate color was, how does it appear, what was the colony type, going back to the O antigen serogroup screening, then to the IMS. So these two steps will also then decide, is it do you need to continue test or report as not detected? Now within SynBio, we call this point as exit point. Why exit point? Because if you see from this flow chart, it has got multiple other steps involved as part of confirmation too. But if your test stops here, client doesn't have to pay for all the steps, and this is just ethically, we believe, is not correct for us to charge you for something that we are not even performing. So we have got different, models for pricing here. So we can just say you just pay up to the exit point. If your test stops here, we report the results are not detected. You don't have to pay for the next steps. We don't have to perform the next steps. But if the testing has to go past the exit point, that is, you're getting typical positive reactions as expected, then we continue for the whole confirmation process. Now, the next step is called MALDI confirmation. MALDI confirmation is a term that's again very widely used in industry nowadays. MALDI is a technique used to be in chemistry. It's a proteomics based technique where you have your sample, you have your organism. Each organism has got certain protein biomarkers on their surface. In this case, you then put this sample through certain techniques, and you put into an equipment where it produces a footprint of different proteins present on their surface. That footprint of proteins is then aligned against a library which is already present in the MALDI machine. And then once it aligns, it tells you, okay, your sample has got A protein and your library has got A protein as well. And both of these are coming from E. coli. So this is how when it aligns your protein from the sample against the library, it gives you identification of the organism. Now this method is an AOAC method widely approved by DAF and industry. And then once you have done the MALDI confirmation, it will help you identify that the isolate that you are targeting right now, is it actually E. coli or not E. coli? Now there could be chances that it's actually not E. coli. So if the MALDI doesn't confirm the identity of this E. coli, the result will still be reported as not detected. But if it has gone past the MALDI confirmation stage, then you have to go back to the third stage as well, which is now PCR confirmation. Now, this PCR confirmation is the same confirmation that you have done at the screening stage. However, in this case, the confirmation is saying that is this STX and EAE genes, are they actually coming from the viable isolate that you have on the plate, or was it just from the dead cells in there? Now, if there are chances that the organism that you have isolated may give you very typical phenotype on the selective media plate. It can even go through the antigen screen. It can even confirm through MALDI. But if it has lost the STX and EAE genes, you have to call the result as negative. So that still means that if it doesn't have the genes responsible for producing toxins, the results will be reported as not detected. Once it has gone past all these this test and has confirmed positive at every step, this is when the results are confirmed positive. But if the sample has given any negative result at any step, we just don't stop there. We have to keep going back and confirming the other isolates present onto the plate as well. So that's one thing that if you don't get, for example, we took one isolate, we did O antigen screen, it came back negative. We can't just conclude that all other isolates on the plate are also going to be not detected. So by complexity of the test, I mean that you have to keep going back and be very, very confident unless you can definitely say that all the isolates that you have, they are not detected. You have to continue performing the confirmation. So the time it takes to do confirmation may go from 2 days. If it's quickly first isolate confirmed, we can conclude in 2 days, but it may take 3 to 4 days if we have to keep going back to these isolates to get a confirmed positive result. Next, please. Now, once you have a confirmed positive result, this is how the test report will look like. Now, a confirmed positive result, what does it mean? It actually means that the isolate that you have identified is actually E. coli 0157 or non-0157, depending on the serotype identified. Now, this isolate also have got Shigatoxigenic gene, which can cause illness. So the results are coming from the first part is the PCR screening part. Then we have done the confirmation, and now the result that will report to you will be called confirmed positive. And we will also give you the identity of the serogroup that's been identified in here. Now, what you need to understand is that you need to take appropriate action as per your HACCP plans, as per your regulatory requirement, or as per Department of Health guidelines. As I said, this may take 2 to 3 days, but if you look at the test report that we have in there, the first circle which says potential positive and confirmed positive. This is where majority of the questions come to us. Now, this whole industry for STEC testing, as I said, is widely regulated by DAF, Department of Fishery, Forestry, Agriculture. Now, this DAF requirement are again regulated by meat export testing, and it's a global requirement. So the reporting that we perform has to be aligned with that. I understand when it comes to local Australian market for leafy green, this may not be the requirement, but because all our reporting, everything is assessed by DAF and NATA together. So we try to keep single reports so that, it can serve both purposes. Now, in this report, you will see that when we have a potential positive screening result, we are asked not to remove that. So even if your results are confirmed positive, you will still see the first primary screening result appearing onto the report. So the first line here, which is GDS Big 6 STEC screening, it gives you the method and it also tells you the first primary screen is potential positive. And then we have done O serogroup STX EAE testing, and that is confirmed positive. If you look towards the bottom of the report, it gives you definitions that we apply for you for you to have good understanding that what does the results reported here means. So confirmed positive is E. coli O111. So that is the identity of the serogroup that's been confirmed in this testing. So when you're looking at the test report, these are the three components you should be paying attention to: your screening result, your final result, and what kind of E. coli serogroup was identified in there. So this is how your complete report should look like. Next, please. Okay, now there are chances, as I said, at any point, if we go back, when we discuss the confirmation part, if at any steps during confirmation, your sample has returned a negative result. So it's not the screening result, but anything during confirmation has come back as negative. Then the result, the final result that we report to you is ND, which means not detected. How you should interpret that? Your STEC is absent in the sample. You don't need to take any action, and it would have taken 2 to 3 days after your first potential positive to receive this report. Now, in this report, what is critical, again, the same question, we cannot remove your screening result. That will still appear as potential positive. So your final result is, although not detected, but potential positive is not your final results. It's referring to the screening. So if you look on the left hand side of the report, it still says GDS Big 6 STEC screening. That is potential positives, and it's not your final result. Then you have your 6 STEC O serogroup STX EAE result, which is not detected, and that is your final result. So ND, again in the definitions, we will tell you that this E. coli species has not been detected in the sample. Now this is very, very common. Sarah also touched base on this one. When you're packing your product in your warehouse, you're using some kind of bleach solution, some kind of wash solution. That means you're getting rid of E. coli in the sample, right? So if you have gotten rid of the E. coli there, or there could be some, it's called free DNA. Free DNA is the DNA from your dead cells. Now PCR being such a sensitive technique, it will pick up those DNA fragments, it will give you amplification, and it will give you a potential positive result or a PCR positive result. Similarly, go back to your COVID dates when people were saying X person has been identified COVID positive but is not infective. So same example here, the sample has been identified as potential positive but doesn't have the E. coli to cause infection in here. So same principle applies in here. So it happens that when we are doing confirmation, for example, if we're getting 10 samples for confirmation in a week's time, it could be only 2 or 3 samples will confirm, or that gives you 70 to 80% of the samples which are PCR positive at the screening stage will not confirm, and it's absolutely fine. At least the idea of introducing or implementing this methodology by department was it's giving you a quicker turnaround to screen the negative samples. So at least you're not holding up your whole shipment or holding up your food with short shelf life for 5 days before you can find out the sample is positive or negative. So at least you can perform a quick screen. If samples are negative, no drama, go and sell your product. But if the samples of those samples being tested, if some of these come back with potential positive, you have to wait for the confirmation. And just adding on in here, when we perform testing in the laboratory, when Sarah's lab will perform the testing, if she has run, say, for example, 30 samples for PCR screen today, she may not get any sample PCR positive. So this is a very good advantage of quicker turnaround time for PCR. So it has got its major advantages as well. So when in our laboratory, for example, in a week, we may get, say, 3 samples from, say, Melbourne laboratory, which are PCR positive out of the 100 sample that we have tested. And out of the 3, only one may confirm and rest may not confirm at all. So this just to give you a statistical idea that yes, this has got advantages, major advantages. The only disadvantage is that if you get a potential positive, you have to wait, and after 3 days it comes back not detected. We understand there's a frustration, but that's how the whole testing works. This is very regulated industry. Next, please. Now, when you're looking at your test report, what is very, very critical for all of us to understand is that, again, the reason we have put up this particular presentation is that we had many clients asking how to look into a test report. How do I find that what you are reporting is correct? How do I have confidence in the test result? So the first important thing is that when we are looking at any test report, you need to find your NATA logo. So NATA gives you peace of mind. NATA gives us this scrutiny that, oh, because we are approved by NATA, we are accredited by NATA, we are following an ISO 17025 quality system, we need to make sure we have got right systems in place. That could be set from your, published methods, from your reporting, from your staff training, for maintaining competencies or maintaining other process controls within the laboratory. How do we review the result? How do we report the results? So everything comes under the umbrella of ISO 17025. So we as the labs must comply for that. We also have our internal audits that make sure that laboratories are complying with the NATA requirements. Now that is just part of this testing. When it comes to meat export testing, we also have to align with DAF requirements. So when we get a NATA audit, DAF department also comes along with NATA. They also audit the lab. They have got separate checklist on their own. They will make sure we are following the right method, we are following the right technique, we are reporting in correct way, we are interpreting the result in the right way. We have got right methods and procedures in place. So there's a lot of systems that are required, a structural framework that is required to maintain NATA and DAF requirement. That's your first stamp of approval that gives you this confidence that the results that we are producing are correct. Now, the next thing is, if you look on the top of the report where it says certificate of analysis, the circle there highlights the certificate number. So when we give you, issue your first report, you will have a certain unique certificate number, and in the bracket it will say R00, which is the revision number, which means that this is the original test report that we are giving you. We haven't made any revisions on the test report. This is the first test report that will go out to you. Now, this is important because there will be times, as we discussed in the next slide or so, that sometime it will have R01. That means it's a revision 1, or R02, revision 2 of the test report. That means something that's been issued you at the initial stage has been updated. Some amendments have been made. So based on the number of amendments, you will see this number R01, R02 onto that. So this is for your lab staff, for your internal quality team to understand what does the certificate number means in here. Now, this is the basic things that you will see in any report. Now, let's talk about the results in particular related to STEC only. Now, first thing that you need to see is that it will have certain reporting components. So your test report will tell you, are you testing this product for E. coli O157? Are you testing it for big 6 STEC? That means O157 plus 6, that means the top 7. As I said before, meat export may have different requirements. Sometimes you just need O157 testing. Sometimes you need O157 plus 6 STEC. So depending on that, it will tell you which analyte. And then most importantly, it will tell you, is it screening or is it confirmation? So if you look on the analytical result, the circle on the left-hand side is telling you E. coli O157 screening and then big 6 STEC screening. So these two results are applicable to the primary PCR screen done in the laboratory. That's it. And then important thing is method. you should be able to see which method the laboratory would have used on your sample. Have they used any approved method? Is this method approved by department? Because as I said, department has a checklist. You can only use certain methods which they approve, and they accredit the lab to use those for. And this should be very clear, because if you go and look into, say, MLG5CO3, this method is publicly available. So you can select the method, you can find out the details, like how did we process a sample, what did we checked into, how much did we weigh, which enrichment media did we use, which equipment did we use, how did we interpret the result. Uh, Similarly, if instead of MLG5C there was some other AOAC method, you will be able to go, if not publicly available, you'll be able to buy this AOAC document. This will give you all the details on which matrices was the method validated on. Is this method approved by by DAF? What covers in this method? What is the enrichment media? How long is the incubation? Which genes are you looking at? Which platform are you using? How do you interpret the results? So this gives you this information and transparency, so that you are clear as part of your own regulatory compliance, and otherwise that which method we are using for your testing. Then are the units. Now, when you send us samples, you may just need 25-gram testing, or you may need 125-gram testing, or in the case of an outbreak, we may need a composite of multiple subunits, and it could be even 1,500-gram testing, or for meat export, it could be just 375-gram testing. So this unit is again standardized. I or the lab cannot decide, okay, we are going to test today 20 grams, or client only gave me 100 gram, I'm going to test that. This is very defined, and it all depend on your sampling plan. So this will, you have to go back and look into Fasan's compendium and otherwise, and decide what your sampling plan is, how much sample you need to send to the laboratory for testing. So these are the other important components which you need to see onto the test report. Sometimes client may just say, okay, in terms of outbreak, I need to test multiple units separately. So that's like a special case, and you will have some sort of a logic behind it. Maybe Department of Health have asked you to go and sample from different site and test the whole sample. That can be accommodated, but it must come with certain logic that why we are doing that testing, if for you and for us as well. Now, the results in both these cases are ND, which means not detected, and then we go from there. Next, please. Then we, as I said before, we have got two-stage model for confirmation. So the first one is we done screening part. Now this report is referring to a confirmation report. This is for an E. coli 0157, which must have been presumptive positive at your screening part. Now the units in this case, if you see, are per broth. That means now we are referring to the sample taken from the enrichment broth, not to 375 gram, not to 25 gram. And this could be the broth was sent to us by one of the abattoirs, which they have, where they have done their own primary screening, and they vote SymBio to confirm that if it's a true positive sample. Now, when we would have got the broth, first thing we did was IMS step. And if the IMS has got growth on it, it's called still potential positive, because it still has to go other steps to confirmed on. The first result that we report is called potential positive. Now this is gone past the exit point. So then we go and add other tests, which is STX, EAE, and MALDI confirmation. Then we perform those tests, and this has returned you a confirmed positive result at the end. So what happens in this case, if we have a broth sample which was potential positive for screening, you send it to us, we did the first step of confirmation, potential positive again, we confirmed using different techniques, and it has come back confirmed positive. And if you look at the bottom of the test report, it tells you that confirmed positive means it's an E. coli 0157 has been detected in this sample. So this is giving you a whole idea that how a confirmed positive report should look like. Next one, please. I see there are some questions related to confirmation. Once I finish this one, we'll come back to it and explain in detail your questions on that. Now, for confirmation step within Symbio, we've got two methods which are approved by USDA FSIS, accredited by NATA, and they are approved by DAF as well. The first method is E. coli 0157, MLG 509 method, and the other one is MLG 5B05 method. Now, the E. coli 0157 is only applicable to E. coli 0157, and non-0157 covers all 6 different serogroups. So we have listed Symbio method numbers as well. As we mentioned before, all Symbio facilities are accredited and approved to perform screening method, but confirmation is performed in our Symbio Brisbane and Melbourne laboratory only. So because again, we need to maintain the competency, the laboratories participate in external PT programs with IFM, and also internal interlab programs within Symbio, which is set up by our internal QA system. So IFM will send us blind samples spiked with genetic material of O157 and non-O157 different serogroups, and we do this testing twice a year at both laboratories. The comprehensive testing takes place and the reports are sent out to IFM for assessment. IFM will make an independent assessment and let us know if we have passed or failed on that. So this is more like process control that we have in place. We also, when we set up this testing, we also have got our laboratory positive strains tested along the process from start till the end for PCR screening and for confirmation as well, which gives us this confidence that the results that we are producing are right and are valid. Next one, please. Okay, now coming towards the conclusion of these test results, that what do we understand from presumptive positive test result or confirmed result? So going back to the slides where we discussed the screening of it. So presumptive positive result is only a screening result. It cannot be considered a final result. And what does it indicate? It indicates that STEC may be present, the Shigatoxigenic E. coli may be present. It's not confirming that, and all that it's telling you that in the sample that that you have sent to us, the one which we have analyzed has got STX and EAE genes present in there. Now these genes can be can be present in the non-viable E. coli cells as well, or the cells that cannot be cultured. There are different terminologies used within the industry when we say, okay, they are viable but not culturable. So if we cannot isolate that E. coli, the final result that we have to report is actually not detected. So STX EAE presence only does not conclude that you will have in E. coli that's going to cause that may cause infection. Now the free DNA present in the sample can also give you a positive PCR result, which we have to report as a presumptive positive to you. But once we have done the confirmation on the sample, if your sample has been confirmed, that definitely means that your sample have got E. coli. E. coli isolate, which is now confirmed to carry the pathogenic genes STX and EAE, and we were able to isolate that E. coli. That means this E. coli is living. That means if it enters human system, human gut, it may be able to cause illness or infection because those genes are present. That means if it gets a right trigger, it may produce the toxins as well, because all that it's doing, that these genes are capable of producing toxins. And if it does, the confirmation then gives you the right measure. The Department of Health can take action, the supplier can take action, or the grower can take action. So the confirmation part is very, very important for you to make the right decisions on right time. Now, before we conclude on this one, I just see that there was a question regarding Shiga toxins, like what are they? These Shiga toxins, they are, I did mention that E. coli gets killed, but here we are not referring to the detection of toxins. This forum and this testing is for Shigatoxigenic E. coli, the E. coli that produce STX EAE genes that have the capability to produce toxins. So we are talking about E. coli that can be killed at 70 degrees, but the toxins, they are heat resistant. They will not get killed in here, but the E. coli producing these toxins will get killed. So that's a separate topic on its own, that yes, the toxins remain, toxins are heat resistant. They will not be killed. We are only referring to E. coli in this forum. And then there are, various publications where they say that if you do the pasteurization or if you do the heat treatment above 100 degree, it may kill the toxin. It may damage the toxin, but again, there's no sure thing for us to make a claim on it, especially related to this forum. Next, please, Kristen. All right, so now we are officially open for the questions. I'll quickly go through some of this, if that's okay. Kristen, can I go through some of the questions in the chat now?
Yep.
Okay. All right, so the first one, again, we've just discussed that we're not referring to toxins in here. We are referring to the genes that produce toxins. So yes, they are resistant. They are exotoxins. So completely agree with you. Then plans to implement confirmation. Yes, we have. We've got NATA and DAF approval for confirmation at Melbourne Laboratory. We're just waiting for official letter. But if you want to get more information, please get in touch with Kristen. He will have more information on it. The lab is actually approved for DAF for screening. All the SynBio laboratories, they are approved by DAF for verification, for screening test, but confirmation is at Brisbane and Melbourne starting now as well. Next question is from Michelle asking, is there a difference between presumptive positive and potential positive? It's the same thing. All that it refers to is PCR positive, but DAF want us to report any PCR positive as potential positive. So this is the terminology which is regulated by department, and they want, in this case, results of PCR positive to be reported as potential positive. If you're talking about salmonellas and other, say, listeria in dairy industry or otherwise, the lab usually report as presumptive positive, not as potential positive. So this is something which is very STEC related by DAF. Then we have another question. Okay, identifying of serogroups in screening tests. Yes, we have. Again, it's a PCR screening we are referring to. It's not the confirmation result here. So the method that SynBio uses is MLG5C. That's our preferred method of testing, and this method has got two stages of testing in there. The first one is we are looking for STX and EAE genes. If the STX EAE genes are present, we are not reporting anything to you. We are going for O serogroup screen. At O serogroup screen, we are looking at all the non-O157 STEC, different O serogroups, plus O157 confirmation. If that PCR comes back positive, then we let you know your sample is potential positive, and we proceed with confirmation. If that O serogroup screen is negative, we conclude the results as negative at that stage. So this is an additional layer which MLG5C method provides us, and we are not issuing you any report, because if you look back in the days, we have that technology as well, and we're not against anyone, but there are certain platforms like, for example, BACS. So in BACS, if you're doing first screen, STX EAE, sample comes back positive, lab will issue a report saying it's potential positive. Then they will go back and do the screening for, it's called panel testing. They're looking for the serogroup. And if it's serogroup negative, then they will go back and say, oh, actually your sample is negative. Whereas in MLG5C, this step is already included in screening. So you don't have to wait for next day to say, oh, the serogroups are not present, so we are not proceeding. So it's covered in there. Now, mutant bacteria, selective media can be, look, again, that mutant bacteria, this is a topic that is more like a research based. When we're talking about STEC testing for top 7, everything from sample way up to the confirmation and reporting is regulated by DAF. So even if, we know that mutant bacteria can grow, cannot grow, we unfortunately cannot have any input into this one. We just have to follow what DAF or FSIS is asking for it, because you have to consider these samples are going to US or Canada. If we start making these decisions here that, oh, it's a mutant bacteria, and Canada says, no, it's not, you're talking of two countries talking at a diplomatic level of this kind of, if there's a recall or if there's an outbreak or things like that. So this industry is heavily regulated. We cannot even move an inch from what the method says. We just have to go by. They ask us to use certain enrichment media, we have to use that. They ask us to use a certain plate for this one, we just use that. So interpretation, whichever way DAF will ask, whichever way FSIS will ask, we just go by that. So I get your point.
Can I add this point? Sorry, I'm Kashif Ahmed, I'm the operation manager for this question of mutant bacteria, and also it's good to basically expand this thing to the other testing and other method as well. We always said this thing, and just to understand that when the test is done, the test is done as per the definition and the scope of the method. So, what example people will say that whatever the salmonella can get mutation in the environment and is not really basically cannot be detected because it lost some of the genes which can be. So the good thing is that basically the testings are done as per the scope of the method. It's not what the lab is really want to do or not do extra. It's a good thing as well. I'm also giving an example that if a salmonella test is done for 25 gram or E. coli 7 test is done for 25 gram, what about basically if somebody do 50 gram? So the fact is that if it says 25 gram, just do 25 gram because this is the sampling plan, this is the regulation, and this has to be done as per the specific method, whether it's Australian standard, whether it's AOC method. So that is basically the answer that if anything mutate, it could be any bacteria and other testing as well, but those mutation is not basically the scope. But there is one thing we can tell you about the E. coli 157 STEC as well. When this start something from like 25, 30 years ago, the focus on serogroup is much more as compared to other testing like salmonella, which means that the definition of STEC is also changed in last 25 years, depending of what gene is carried. So the latest definition is that it has to be STX and EA genes. And the serogroup as well, for example, some serogroup are not found in Australia, but they are found in other countries. So US FDA or other European countries and Australian, basically, the Department of Agriculture, they're also monitoring even the presence of a specific serogroup in different countries as well. So this area is very good, the consideration of different serogroups and those things as well. So I think it's good, but at the same time, all the testing, all the targeted bacteria, it has to be tested as per the scope of the test method, which is approved for this testing, and that's the way we do it. Right? All right, I think that will clarify more. Right?
All right, so, and then there's the question regarding the difference between M8.10.4 and 8.10.7. See, these are, when we say M8.10.4 and 10.7, these are internal SynBio numbers, and they refer to what kind of testing you are after. For example, as we mentioned, sometimes you just need E. coli 0157 testing, sometimes you need all the top 7 testing, and then depending on the test requirement, you may your compliance is for certain AOAC method, or you're asking for MLG5C method. So based on that, we have got different numbers. M8.10.4 is actually our GDS method, and M8.10.7 is actually a confirmation method for MLG. And then if you need more information, again, please reach out to Christine, to myself, and we can give you more details of the different SynBio method that you're currently using or you are interested to use in future. In terms of sampling for a lot of presumptive positive sample, Kash, if you want to answer this question, what advice can SynBio provide if there are a lot of presumptive positive samples?
Can you guys also click the chat portion so we can see the direction, right? Okay. This one basically, which one is that? What is that? All right. Would you like to do more sampling from a lot of presumptive positive? But the first advice is that presumptive positive result should not be used as a conclusive result, especially in the E. coli 157 STEC role, because this result is presumptive on a very strong term. I'll give an example that if it's required to find the STX-EA gene, you will find E. coli, which has only EIA gene, and you find another E. coli in the sample, which only carry STX gene. In the screening method, they will provide a presumptive positive. It will detect gene from 2 different bacteria. That's the reason it is presumptive positive, right? When the confirmation is done, in the confirmation, it will find out, is there any bacteria, E. coli, is there any E. coli that carry both genes in it, plus which serogroup it belongs to. So the confirmation of E. coli 157 STEC is very, very important thing. Any action should be taken based on the confirmed result, not based on the presumptive results, right? There is a practice, if you compare this practice with Salmonella and Listeria, yes, there are practices that some food, especially manufacturer, they actually take some decision based on presumptive result of Salmonella and Listeria, because there is one thing there, right? If we, for example, somebody use a screening method for PCR and provide a presumptive positive for Salmonella and Listeria, we can tell you that it is 99.9% chances that it will be positive on the confirmation, right? In terms of STEC and E. coli 157, just a rough idea I can tell you, if we test 1,000 samples, it may be 30 or 40 samples, it will be presumptive positive, and then maybe only 1 sample will be presumptive after the confirmation, right? So that is aesthetics, right? Now it is your call. Generally, if the testing is done for meat export, they have a very clear requirement. There is a presumptive positive is just a result to say that is required further investigation. In fact, this result also reported to USFDA as well. Everything is there, but the whole everybody understands that presumptive positive doesn't really mean positive. In terms of other products, for example, juice or leafy greens or other products as well, our recommendation is that presumptive positive should not be taken as anything really serious, unless there is something you want to decide and you have a history from your results in the past that when you have a presumptive positive, would it turn out positive as well, then you can make your decision. In a simple word, presumptive positive of STEC E. coli means nothing really as such. Is a very high chances that the confirmation test will conclude that it is not detected. All right, so that will say that's the reason sampling will come as well. All right. Okay, Lovely, you want to go to the next one? I can close. Can we retest a confirmed positive when a confirmed result is disputed, right? It go back to the point that why a confirmed result is disputed. From the lab, if a confirmed result is provided, there is we can tell you there is no need to dispute that result, because a lab has done a lot of things right. We have done the 10 PCR on one sample. We have done the serology, IMS and everything, and multi-TOF, which means the confirmed result is truly confirmed, right? You may say that, for example, your result is coming from a product which was incidentally was in contact with some high-risk area, and you may go back and do more testing to confirm that the lot is still clean. You can do all these things, your decision for your sampling plan, but we're not gonna say that excluding a confirmed result just for that purpose, right? Okay, so I think it is not really good. Kind of say when we dispute the confirmed result, right? All right. The next question, right? Doesn't mean that all the confirmed positives only declared after successfully asked. Yes, this question is very clear. Yes, it's from no, right? Yes, the confirmed result, as Loveleen also explained that. As we said that presumptive positive is from the population of E. coli. When we do, first thing we do the panel testing and initial screening to say that which group is possibly belongs to. The lab run the IMS with the immunomagnetic separation things to target the bacteria, the extract level of extractions. Then it grow on the fly. Individual isolated spec is done the serology for the sero group. Then the individual isolate is done PCR, right? So normal real-time PCR is only one test right here. We're doing 10 real-time PCR on individual isolates. So please, it is really done after all the identification and confirmation, right? Okay. Okay. Is there any possibility to get confirmation contamination of sample with E. coli? With E. coli, with E. coli, right? Okay. In general, yes, E. coli is that, for example, I will extend this question to E. coli 157 STEC. I will just say that way. So in terms of the people hygiene, because the STEC is not commonly present in the with the human beings, right? It is unlikely that any personal hygiene of the lab staff can create cross contamination, but the lab can have the cross contamination um, um, at the testing stage. It is very unlikely because the test samples are way in the different area where there is no positive controls. At the PCR stage, there is a possibility that if there is any splash happen from positive control, it will be done. But because of the basically it is a genetic testing, there are a lot of basically further detail of checking imperfection control and a positive control, negative control in the glands. So the lab is able to detect if they have done any contamination. It frequently is not happen, but if we see something like that, right, that let's say, I'll give an example that we run 10 samples and one is positive and negative control is negative and positive control is positive, it's okay to accept it. But if we run 10 samples and all 10 are positive, even the negative control is negative, we say, look, there is a doubt that how come the 10 sample is positive. We have a history from the client as well that don't have any positive. We as in biolaboratory, we do the retest in this case. We don't simply accept those ones. So yes, there is a chance, but the chance of having STEC environmental contamination is very rare because this organism is not really present everywhere. We mentioned, okay. All right. So what is that basically with the recent recall in the Spanish over STEC, how much testing sampling is required by an organization, right? Is there a guideline promoted by DAF that tells about the frequency of testing? DAF may not say anything on that one because DAF is more, there is a compendium available for is for testing, and in this compendium for it literally has a chapter for basically section for E. coli 157 STEC as well. It also provides some guideline. It basically second thing is that it also go back to the point that what it was actually the sources of basically the is related to the specific Spanish area. So it could be something, but I can tell you there is no clear guideline available from that. It's just a compendium of Fizan. It will provide some actions for what should be taken. And they also talk about the testing of zero group and other things as well. So yeah, that's you. Is there any possibility to get contamination in the sample with handle the sample in your lab rather than sample in your laboratory? Yeah, as I already explained before, right? Because the equivalent 57 STECs are not commonly present in the environment of the lab and with the people, the chance of contamination is less. But yes, it can be possible. But we, as a sensible testing laboratory, we make sure that we actually do the retest as well. Sometimes we can tell you basically. Sometimes we find very high level of peak basic in the PCR, and it can be called presumptive positive, but we rerun it and we make sure that the peak is really the true peak. So all those are things we do in the lab. Okay, is there a limit on the number of colonies that should be confirmed for E. coli 157? Because E. coli 157 is only one isolate, so the rule to test 5 colonies to take in confirmation. For STEC, because it's a bigger group, there are 10 colonies taken for confirmation for STEC. Okay. All right, the next question is that this may be unrelated, but it's the process of screening test with the presumptive positive way. You can then conduct a confirmation test done on other pathogen bacteria like Listeria or Salmonella. We don't do that. Usually it is done by the pathology lab in Medicare. You ask for the blood test for sugar. They may also test for LFT as well. Now in the lab, as I said, that we are bounded with this rule that if you ask for E. coli 157, we only test for E. coli 157. We will not test your sample for any other bacteria. I can also give an example that in our lab, we have a multiplex Salmonella and Listeria method within the multiplex, right? But if your testing requirement is salmonella, it will be only test for salmonella, even if we have something for listeria as well. If you come back and ask that, is there any way to do listeria testing? We may tell you that we have a multiplex, your data is there. We can conclude a result from the data. But this requirement for lab is that we only test. This is also one good thing to mention. It's a good example for listeria mono. And you can see that the different health authorities, in different states have a requirement for pathogen notification for listeria, especially listeria mono. So let's say, for example, in Victoria, there is a requirement to report listeria mono. But if a client is asking testing for listeria species, we do a listeria species testing. We don't go any further for listeria mono because it is not requested, it is not required by. So that is one thing the lab has to do it. They need to restrict basically for the testing that was required to do that. We will not go to any other testing unless the client specifically asks to go further. Okay, so nobody answered some of them, okay?
Yes,
Is there any more question? I think that's all.
I think that's pretty much. If there are more questions, feel free to reach out to myself, to Christine, and we'll be happy to answer your questions. If you need any further information, we're always here.
Excellent. Thanks, Laveen. Thanks, Kashif. Thanks, Sarah.
Pleasure.
Yeah, like Laveen said, please reach out if there's any further questions. And if anyone's interested in a lab tour, we're always open to show customers around. So please reach out again as well. Hope everyone got some good information and enjoy the rest of the week. Thanks, everyone.